Hello, everyone, and welcome to the BC Showcase as part of Bio Boston. Before we move on to the program, I would first like to go over some housekeeping notes. All attendees have their cameras and microphones turned off so as not to disrupt the presentations. If you have any questions for today's speakers, please do so using the Q&A feature located at the bottom of your screen. Here you can type in your question and you can upvote other questions that have been asked by other attendees. In this area, there is also a chat section available. We invite you to use this feature. Please tell us where you're from, what excites you most about today's event, and anything else you would like to share. Our speakers and the LSBC staff are monitoring this area and would love to chat with you. If you post your attendance to social media, please don't forget to tag us. Our handle is lifesciences_BC and our hashtag is #LSBC events. Without further delay, I'm pleased to welcome Wendy Hurlbert, our President and CEO here at Life Sciences BC.
Thanks, Claire. Welcome, everyone. We're very happy to have you join us today, and wonderful to see the interest in the BC delegation that is on its way to Bio. As we begin, I would like to acknowledge that I'm joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that others are joining us from other traditional and unceded Indigenous territories across this beautiful province and country. For those of you that are not familiar with Life Sciences BC, we are a not-for-profit association. Our members consist of academic and research institutions and local and global companies operating in the life sciences space. For example, digital health, medtech, med devices and diagnostics, biotech and therapeutics, and those companies that provide services and support to the broad life sciences ecosystem. We're funded through membership and the generous contributions of our sponsors and partners. As this is the Bio conference, what you're going to see is a great selection of some of our biotech companies, and hopefully you'll come and visit us while we are in Boston. This event is going to showcase their innovation taking place, and we hope that you're going to enjoy hearing from them. And at the end of this event, you'll see a QR code that will take you to our booklet where you can see the full scope of our delegation that is coming. We'd like to kick this off. We're super excited to have with us to start our programming, the Honourable Brenda Bailey, Minister of Jobs, Economic Development and Innovation with the Government of British Columbia, who will be sharing a few words. As a small business person, tech entrepreneur, and change maker, Brenda Bailey was elected as an MLA for Vancouver Falls Creek in 2020 and named British Columbia's Minister of Jobs, Economic Development and Innovation in December 2022. Minister Bailey brings to her role a passion for expanding opportunity and economic innovation, nurtured over a lifetime of building new businesses, leading economic development initiatives, and serving her community. In every role she's taken on, she's worked to create a more inclusive community and economy. And I can tell you, in her short time as the minister, she's had a massive impact on the life sciences sector. She's also blazed new trails for women in tech as a co-founder of Canada's first woman-owned and operated video game studio. She helped strengthen and diversify BC's creative tech industry as head of British Columbia's Interactive and Creative Digital Marketing Association, DigiBC. Today, as the Minister of what we like to call JEDI, her focus is on working with businesses, Indigenous and community leaders to drive the government's Stronger BC economic plan forward. For Minister Bailey, that means growing an economy that benefits all British Columbians who support for business, expanded access to post-secondary education and skills and training, and meeting our climate commitments and fostering innovation across our economy. With that, I'd like to turn it over to you, Minister Bailey, for the opening words.
Great. Hello, everyone. Thank you very much, Wendy, for that introduction. I, too, would like to acknowledge that I'm speaking to you today on the unceded and traditional territories of the Musqueam, the Squamish, and the Tsleil-Waututh First Nations peoples. As the Minister of Job, Economic Development and Innovation for the province of British Columbia, I'm really excited to get a chance to talk to you about our life sciences sector, because as many of you know, it's been an exciting, fast-paced time for our sector in 2023. But of course, it's always an exciting time in life sciences because of the life-changing, forward-looking, innovative work our talented researchers and companies are involved in on a daily basis. I've had a look at the lineup of speakers that you'll be hearing from today, and it's going to be an inspirational time. I think about the collaborations, the sharing of resources, the talent, and the knowledge that's taken place between academia, industry, organizations, and government to help establish BC's reputation as a world-class leader to solve global healthcare challenges and to better prepare us, God forbid, for future pandemics and health emergencies. BC is the proud home of Canada's fastest growing life sciences sector. We're working to create the conditions to enable more companies to be able to continue to grow and thrive here in British Columbia. And that's why we recently launched the BC Life Sciences and Biomanufacturing Strategy. It's a key action of our Stronger BC Economic Plan, and it focuses on innovative, sustainable growth. Our strategy was developed in close consultation with industry, academia, other governments, and advisors. And we've had input from the best and the brightest, some of you on this call, to ensure that BC is positioned as a global life sciences powerhouse and a leading center for commercial scale biopharmaceutical and medical manufacturing. The strategy is designed to help innovators, entrepreneurs, contract manufacturing organizations, and larger companies grow their businesses. Through the strategy, we're creating an attractive environment for companies to create more well-paid jobs and to scale up and commercialize, and to create new opportunities to test and manufacture the most promising drug candidates and medical devices right here in BC, so they can export and partner globally and save lives around the world. We're making it easier for innovative treatments through clinical trials to take place here in our province. And we're strengthening our connections to the global life sciences supply chain and international markets. We also know that with growth comes the need for skilled labor. Earlier this year, we announced with the federal government an investment of more than $7 million to create a new national biomanufacturing training center, where we will train up to 700 trainees every year in leading edge industry recognized curriculum and equipment to help companies in BC hire skilled, talented people and continue their important work. I was also pleased to join Premier David Eby earlier this month as we announced our $482 million Stronger BC Future Ready Action Plan. This will help thousands of people get the skills that they need to succeed in our changing economy. We'll be making post-secondary education more affordable, more accessible, and relevant to British Columbians now and into the future. We know that early stage companies also need access to affordable, high demand, and specialized lab space, and we're developing more here in British Columbia. That's why our government has invested $12.5 million to wet lab and biomanufacturing facilities with Admare Bio Innovations in Vancouver and with Vancouver Island Life Sciences in Victoria. These are exceptional visionary partners, and their expanded operations will help early stage biotech companies to test and to grow. As a province, we offer many advantages for you to choose to start your business here or to invest in local companies. We are actively working to create an economic investment climate that is environmentally, ethically, socially, and economically conscious. And we've grown into a global leader in the ESG space. With the Clean BC Roadmap to 2023, we're accelerating climate action in this province to meet our climate targets and to build a cleaner, stronger economy. We offer competitive tax rates, incentives, and other programs that make sure business can continue to grow. The natural beauty is also out of this world and provides a wonderful lifestyle. Let me close by saying British Columbia is working hard to help the life sciences sector so that discoveries can move from bench to market to bedside. Our government is committed to putting people first, to helping BC companies scale up and commercialize here, to create more well-paying jobs for British Columbians, as well as to test and manufacture the most promising drug candidates and medical devices locally, so people can export globally. Through the Stronger BC Economic Plan, we'll continue to build an innovative-based, cleaner, more sustainable, knowledge-based economy that is inclusive and works for everyone. Thanks very much.
Thank you very much, Minister Bailey. We very much appreciate your leadership and your commitment to the sector in British Columbia broadly, and look forward to being with you at BIO.
Thanks, Wendy.
Okay, I'm going to now provide a bit of an overview of our ecosystem. Minister Bailey did a fantastic job of giving the macro environment, but want to give you a little bit of a snapshot of the size of the sector. So if we could move into that. So a little bit about British Columbia, because I know there's a lot of people on the call that are not from here. So we are the westernmost province of Canada. We like to say we're on the best coast, although we're not going to be able to necessarily say that when we're in Boston. So Minister Bailey talked a little bit about British Columbia. We have a population of about 5 million. Our GDP is 3.2%. Our unemployment is around 3.6%. We've had quite a strong economy over the last number of years. We have a very stable government. We have a strong and diversified economy with natural resources, film and media, knowledge economy, and of course, the life sciences sector. And we're home to some of Canada's top universities, University of British Columbia, Simon Fraser University, and the University of Victoria on Vancouver Island. So a little bit about our sector. We are home to Canada's largest biotech, stem cell technologies. They will also be with us in Boston. And we have numerous companies that are over $1 billion in market cap. So AbCellera, Sierra Oncology, which was just recently acquired by GSK, Aurinia Pharmaceuticals, Xenon, Chinook Therapeutics, and Well Health. Again, all of those organizations will also be with us at BIO. We are the fastest growing life sciences sector in Canada, as Minister Bailey stated, with $6.7 billion of revenue, 2.6 billion of GDP, 650 million of exports, and approximately 2,000 companies. We have a highly skilled workforce of approximately 20,000 employees in the life sciences space. That's really based off of company employees. It doesn't include the broader ecosystem of all the service companies that are working within the space to support the sector. And actually, based off of how the data comes together, it doesn't really pick up most of the digital health companies. So we anticipate it's a little bit higher. We've had an extraordinary track record of capital raising over the last numerous years. In 2021, we raised 1.5 billion, our companies raised $1.5 billion of capital, really leading the country, especially based off the size of our ecosystem. So what we're known for antibodies. We have a long history of antibody expertise, which started almost 25 years ago with the spin out of UBC, which is now exploded into approximately 20 companies, if not more, operating in this space. We also, AbCellera, through the pandemic, was actually the first company to discover the antibody, which resulted in the first therapeutic for COVID-19, which Eli Lilly brought to market. We're very strong in regenerative and cellular medicine. There's numerous partnerships internationally where our SMEs and academic institutions are working in this space. Nanomedicines, lipid nanoparticles, our homegrown organization, Acuitas Therapeutics, developed the lipid nanoparticle, which was the core delivery system in Pfizer's COVID-19 vaccine. Oncology has a long history through BC Cancer and numerous companies that spin out of our academic institutions. You're going to hear from a few of them here. Genomics-based precision medicine, gene therapies. We also have a very strong medtech sector in the medical design space, prosthesis, UV and infrared equipment, and a very strong emerging area in 3D printing, which is really tied into that regenerative and cellular medicine space. We have numerous digital health companies operating. We're certainly seeing, as many jurisdictions globally are seeing, that merging of digital health and med tech and devices, and in some cases therapeutics. We also see, as it's happening again around numerous jurisdictions, is the use of AI and bioinformatics as it enters into the drug discovery space. We're known in BC for creating solutions for human health, which combine physics, biology, chemistry into an engineered solution. When you look at a lot of the BC firsts in innovations in life sciences, it's where you see that multidisciplinary science coming together to create a solution. For example, we had the first drug-eluting stent. We have a smart knee and hip that's been created by Canary Medical. And you see that in organizations like Accelerate, where it's biology meets physics, meets computer science and AI. So really, really strong, deep tech roots are one of the cornerstones of our life sciences sector. And we also, as Minister Bailey talked, see ourselves as a point of connection between human health, agriculture, food science, and clean tech. And all of this happens because of our amazing talent that we have. So to sum up, because really what you want to hear about is from the companies as well. We have world-leading, world-class research that is recognized globally. Many people immigrate to BC to continue on their research and start companies because of that depth of world-class research that we have. We learned when necessity struck with the pandemic that we can act very fast. As I said, the first therapeutic came out of BC innovation, the COVID-19 vaccines. We also had the first COVID-19 app, and we had many medical device companies working in creating PPE, ventilators, and respirators, where they pivoted extremely fastly. Our life sciences sector is really grounded in world-class science, resulting in innovation. We outpace the country in company creation, as we have for the last 3 years. We value our global collaboration and partnerships and recognize how important that is as we scale our innovation. And we are known for our collaborative nature within our ecosystem, where we see industry, research, government, regulators, and those that support the sector really come together to create our world-class companies. And with that, I think I'm going to turn over to our first company that you're going to hear from, Dr. Chris Tam, the CEO of Integrated Nano Therapeutics. Chris, the stage is yours.
Thank you, Wendy. Assume you guys seeing my slides all right?
Yep, absolutely.
Good morning, everyone. My name is Chris Tam. I'm the co-founder and CEO of Integrated Nano Therapeutics, or INT in short. I'm very excited to be here to talk to you about lipid nanoparticle, or LNP, and how we use LNP to treat autoimmune disease. INT is a preclinical stage company in Vancouver. We are also a spin-out from the UBC, in fact, from Peter Culler's lab at the UBC in 2016. We are very strong in drug delivery technology, especially lipid nanoparticles. Our co-founding team has contributed to 5 approved lipid nanomedicines, including the first-in-class LNP siRNA Onpattro, as well as LNP mRNA COVID-19 vaccine by Pfizer BioNTech. We are about 13 people right now, and we are growing. Autoimmune disease happens when our immune system mistakenly attacks our own cells. The current treatment simply shut down the immune system, leaving the patient immunocompromised. They cannot fight regular infections, such as the common cold. This is just not acceptable, in our opinion. In order to fundamentally treat autoimmune disease, we need to not shut down the immune system, but to train the immune system to tolerize the autoantigens that are responsible in the autoimmune disease. So how do we do that? We use LNP. As you know, LNP mRNA is proven to be safe and effective, as shown by billions of doses of COVID-19 vaccine in people. But we don't want that type of immune stimulation in autoimmune disease. So instead, what we do is make an LNP, as shown on the left-hand side diagram, to contain two types of drug modalities. The first one is mRNA still, and the mRNA encodes for autoantigen that is responsible in autoimmune disease. But in the same LNP, we also put in small molecule immunomodulator. So what happens is after the LNP is injected into the body, the immune modulator would tell the immune system to ignore the autoantigen that is expressed by the mRNA. The result is what we call the antigen-specific immune tolerance. And we have data to show that it works. So on the right-hand side, you see blue bars that represent various LNP formulations that has mRNA and immunomodulators. In fact, like from A to K is different immunomodulators with the same mRNA in the LNP. The orange bar shows the control LNP, which is just mRNA alone in the LNP. As you can see on the left graph, we have formulations that can suppress the proliferation of autoreactive antigen-specific T cells production. Next to it, it shows that we have formulations that can increase the production of regulatory T cells, the good T cells. And the graph below it, it shows that some of our formulations can reduce the production of antigen-specific antibody production. These three data sets are the key hallmarks of antigen-specific tolerance. In our opinion, this therapeutic approach is very powerful because it not only can apply to autoimmune disease, but other indications such as allergies, organ transplant rejection, and anti-drug antibodies. For each of these indications, we simply need to replace the mRNA. Antigen-specific tolerance has been attempted by others for a long time, by many others, in fact. But so far, there is no success in the clinic. And we believe because two important factors are lacking, but our technology can provide. The first one is we use immunomodulator to provide strong tolerogenic signal that others don't really include in their therapy. For example, a lot of people have been using autoantigen only. This is similar, in our opinion, to the very basic immunotherapies for pollen allergies or nut allergies, which don't always work. The second point is we use mRNA in our approach to enable expression of one or more autoantigen. This is also important because, as you know, a lot of autoimmune disease involve more than one autoantigen. So in our opinion, with these two critical factors combined, we have the winning formula to treat autoimmune disease. The therapeutic approach I mentioned in the previous two slides is powered by our foundational multi-cargo LNP system. As its name implies, multi-cargo means we can put multiple drug modalities in the same LNP. As I showed you earlier, we put small molecule immunomodulator together with mRNA in the same lipid nanoparticle to induce antigen-specific tolerance. Another important point I want to bring up in this slide is we can engineer LNP to accumulate in cells and tissues of interest. For our immune tolerance approach, we engineer LNP such that they accumulate in immune cells. How are we different from other LNP or other drug delivery platform? We're differentiated in three ways. One, we can load different drug modality in the LNP. Two, we can co-load. We can mix and match, like small molecule and nucleic acid together in the same LNP. And three, for small molecules, we can control the drug release rate. The diagram on the right-hand side shows that there are companies that are very good at delivering nucleic acids, such as Chinook Therapeutics. There are also companies who are very good at delivering small molecules. INT sit in the middle. We can put two types of drug modalities in the same LNP. And it is this unique ability that allows us to develop first-in-class immune tolerance therapy for autoimmune disease. Our business model has two arms. On the left-hand side, it is our internal therapeutic program. We've been working on type 1 diabetes, and we have good data so far, and we are moving into other autoimmune diseases. On the right-hand side is our external co-development arm. So there we've been working with external partners to develop LNP drugs of interest. a drug of their interest, sorry. There we work with others to de-risk our technology, our LNP technology, but more importantly, we also create a revenue to fund our internal therapeutic program. We have been self-sustaining since day one. At BioBoston, we hope to achieve 3 objectives. One, we're looking for partners who may be interested in our antigen-specific tolerance approach for autoimmune disease. 2, we're looking for partners who may be interested in our LNP for their drug of interest or combination therapy. 3, we're looking for investors to help us accelerate our internal therapeutic program in a seed round. This is it. Thank you so much for your attention. Thank you for having me. I'm happy to answer any questions. Please feel free to reach out. Thank you.
Thank you, Chris. What a fantastic presentation. Every time I see you present, there's just that much more progress in what's happening. So thank you very much. Integrated Nano Therapeutics was actually one of the companies that went through our investor readiness program and was a recent graduate company this year. So fantastic. Our next speaker is Jonathan Jafari, the co-founder and Chief Business Officer of Mesentel Therapeutics. Mesentel Therapeutics is decoding fibroblast biology in order to create superior therapeutics. John, over to you.
Great, thank you, Wendy. I think you can see my screen.
Yep.
Excellent. So thanks, Wendy. Thank you, LSBC. So I'm John Jafari. I'm the Chief Business Officer and co-founder of Mesentel Therapeutics. And we are decoding fibroblast biology in order to create superior therapeutics. So just a little bit about fibroblasts in 2 diseases which are particularly challenging. Sarcomas, which is a rare cancer. There's very limited therapeutic options. They are immune cold, meaning that the immune system doesn't recognize them. They're very challenging from a drug development standpoint in that there are, I think, 80 subtypes. And so what we've done is really to bring precision medicine to this rare tumor set by developing novel genetically engineered mouse models for specific subtypes and understanding how the cells of origin develop, what programs are activated, and which programs to target for therapeutic intervention. And so this is our lead program, and we expect to nominate a antibody-based program later this year. So fibroblasts also participate significantly in fibrosis. They produce extracellular matrix. They are very much involved in progressive disease, and it's always been thought that once you go into fibrosis, you cannot sort of return. There's very limited therapeutic options as well. Over 200,000 people in North America die every year from idiopathic pulmonary fibrosis. And we're seeing a significant increase in IPF or pulmonary fibrosis due to patients who have severe long COVID. It's one of the sort of long-term consequences. And our solution is really that we understand the fibroblast biology and understand some of the plasticity with these cells and have identified ways to turn them back into sort of quiescent cells to stop the deposition of extracellular matrix and sort of turn off the turning of your lungs into stone and ultimately death. So what's unique about our validated discovery platform? So we have proprietary genetic models which allow us to understand how fibroblasts develop. And then we combine that with single cell sequencing and omics in order to understand which programs that are actually causing some of the issues, depositing the extracellular matrix, providing inflammation in the context of fibrosis, bringing in the immune system in the context of cancer, which targets are actually shutting off the immune system, and allows us to understand which disease drivers and specific targets to drug. So we combine our approach with using the genetically engineered mouse models with human genomics, best-in-class human genomics, single cell sequencing, spatial genomics, and that allows us to identify novel drug targets and therapeutic options. And behind this, we have looked how fibroblasts develop in a number of different tissues and diseases. So we've looked at 14 different models. And so we have this available for partnering. And this is one of the things we're going to be discussing with companies at Bio in terms of drug target discovery. So in terms of our pipeline, we've had in vivo efficacy in soft tissue sarcomas using a small molecule approach, sort of a tool compound. And we are now focusing on a number of antibody targets, and we expect to advance one lead later this year. We're currently completing some immunohistochem for the targets we've discovered, surface targets. And if they match up in terms of what we see with genomics in our mouse model, we will initiate antibody discovery campaigns. And then in terms of the fibrotic side of things, so we have looked at multiple indications. We've compared fibrotic animal models with normals, so we understand which drug targets to look at, and we're looking at small molecule approaches for that. And this program is a little bit further behind, but we expect to identify lead candidates later this year. And another exciting thing that we're looking at is solid tumors in terms of the tumor microenvironment. So fibroblasts actually can wrap around tumors to exclude immunotherapies, and they actually actively shut off the immune system, turning warm tumors cold. And we have just completed in vivo studies looking at some specific targets that we've identified, and we'll be continuing to validate them. And then, as I mentioned, we've looked at 14 different therapeutic opportunities with our discovery platform, and we will both look at opportunities to develop therapeutics on our own, as well as partner with external organizations, because there's really no way we could move 14 different therapeutic programs in parallel. So our business model is really to develop and commercialize the more rare disorders on our own. If you look at IQVIA's oncology, for example, 60 to 70% of rare cancer therapeutics are developed by small companies. So we think this is a really good space for us to play. For the larger indications, such as non-alcoholic steatohepatitis, idiopathic pulmonary fibrosis, it's option B. We're looking to out-license at specific inflection points for larger upfront and milestones and royalties. And then we are certainly open to a limited number of platform partnerships to leverage sort of complementary data sets, as well as companies that are specifically looking at indications that are sort of outside of our cancer and fibrotic activities. So we are about to embark on raising a seed round. We're going to be looking to raise about $5 billion, and that'll allow us to generate antibodies and complete IND-enabling studies for our sarcoma program, complete target validation studies for fibrotic disorders, complete further development and validation of our platform, including bringing in some external expertise in terms of AI and machine learning. And we have already added some scientific and drug development expertise that we expect to announce once we sign our contract with the University of British Columbia and start our fundraising. And this will also allow us for filing our own initial IP as well as initial corporate development. So the team, Michael Underhill, my co-founder, he's been working on fibroblasts for about 30 years and has developed this novel platform over the past 5 years, and is an early adopter of technologies such as single-cell sequencing, single-cell RNA-seq, AI and machine learning. Is really sort of opened sort of the basket for drug targets for this very difficult to target therapeutic class. Leslie Hill, she's a expert in fibroblast biology as well as tumor microenvironment, and she's been driving a lot of these studies in Michael's lab. Myself, ex-QLT, Novartis, so very early experience, biotech, as well as big pharma experience. And then we've had great partnerships with LSBC and the Investor Readiness Program, Creative Destruction Labs, as well as the Stem Cell Network. And our goal for Bio is really to explore platform partnerships, therapeutic partnerships, investors. I'd say we are one of the few companies that are actively out speaking with CROs. So we've got a number of different CROs we're going to be speaking to that really help us move our pipeline forward. And with that, I'd like to thank LSBC, and I'd be happy to take any questions in the chat. Thank you.
Thanks, John. Great presentation and look forward to seeing you in bio. Our next speaker is Stephen Chalong, VP Capital Development at Microbiome Corporation. Microbiome is a clinical stage biotechnology company developing pravibiznan, and I apologize if I didn't pronounce that properly, the first drug in a new class with multiple modes of activity, including anti-infective, anti-biofilm, and anti-inflammatory for the treatment of rare, chronic, and serious diseases. Thank you for joining us today, Steve, and over to you.
Thank you, Wendy. Can you hear me?
Yep, and I can see your slide.
And you can see them. Terrific. Well, good morning, folks. Thank you for joining us today. As Wendy said, my name is Steve Chalong. I was a founding board member of Microbiome, and today I lead our investor relations and business development activities. Today, I'm going to talk to you about our lead drug candidate, pravibiznan, which is the first in a new class of drugs, which has demonstrated disruptive potential for addressing rare, chronic, and serious diseases. So today I'll share with you how pravibiznan is truly differentiated. Oh, my slides are going crazy here. Truly differentiated from other approved therapies through multiple novel modes of activity that include antimicrobial, anti-biofilm, and anti-inflammatory. We're targeting indications with high unmet need and which represent blockbuster commercial potential. We are developing multiple novel modes of delivery as we're focused on targeted local administration, where we believe we can achieve greater efficacy and safety and more favorable reimbursement by focusing on treatment in outpatient settings. To date, pravibiznan has been safe and well tolerated in 5 successful clinical trials involving over 325 human subjects, while also demonstrating signals of clinical efficacy. And our team is led our development is led by a team with over 100 years of collective experience across product drug development, regulatory, CMC, and commercial launch at companies that include Merck and Seprecor. So as I said, pravibiznan demonstrates 3 synergistic modes of activity. First, demonstrates antimicrobial activity with broad spectrum potency against a wide range of bacterial and fungal pathogens, including multi-drug resistant strains. It also demonstrates anti-biofilm activity with an unprecedented ability to prevent and eradicate bacterial biofilms that are known to be a major contributor to resistance in chronic disease, and according to the NIH, are involved in up to 80% of human infections. The reality is that most of today's approved therapies are unable to penetrate biofilms, making this an area of high unmet medical need. And then lastly, we've recently discovered an anti-inflammatory mode of activity in which pravibiznan demonstrates the unique ability to modulate a critical cytokine that is involved in inflammatory and autoimmune diseases. Our drug development programs target conditions with high unmet need and that we believe may benefit from pravibizman's constellation of activity. We have a topical formulation in phase 2 development for the treatment of diabetic foot infections and chronic wounds. We've completed phase 1b studies where pravibiznan was safe and well tolerated and demonstrated signals of efficacy. And we're currently conducting a phase 2 study to further explore this efficacy. And we're also planning a phase 2b/3 study, which the FDA has said may be pivotal. Secondly, we have a local pravibiznan formulation in phase 2 development for the treatment of orthopedic and prosthetic joint infections. In phase 2a studies, pravibiznan was safe and well tolerated at all doses, while also demonstrating a 50% reduction in orthopedic treatment failures compared to placebo. And then lastly, we have an inhaled pravibiznan formulation ready for phase 1 development for the treatment of lung infections. In preclinical studies, our inhaled pravibiznan formulation demonstrated in vivo efficacy against Pseudomonas aeruginosa and another disease condition called nontuberculous mycobacterial lung infections. And it was consistently well tolerated in 28-day GLP toxicology studies. We anticipate starting phase 1 studies in 2024. These indications demonstrate strong commercial potential, which is critical for anti-infective companies. Based upon commercial forecasts that we had developed by Clarion Healthcare, our chronic wounds represents approximately $2 billion in peak annual sales potential for pravibiznan in the US alone. And these are all in future dollars. Non-tuberculous mycobacteria and cystic fibrosis-related lung infections represent $2 billion in peak sales potential. And lastly, orthopedic infections, approximately $1 billion. We also have opportunities to pursue multiple other indications, including inflammatory disease conditions. Pravibismane's unique broad-spectrum potency has been demonstrated by World Health Organization-sponsored studies in which pravibismane and 25 other commercially utilized comparator antibiotics were tested against nearly 3,000 globally sourced pathogens, including multi-drug resistant. In this heat map, green indicates high potency by low MIC values, and red indicates weak potency. In the first column on the left here, you can see how pravibismane demonstrated activity against every isolate tested, and it was the most broad spectrum agent evaluated, demonstrating equal or superior potency versus comparators. And pravibismane's unprecedented anti-biofilm activity is demonstrated here by minimum biofilm eradication studies that were conducted by Dr. Matthew Parsek, a leading biofilm researcher at University of Washington School of Medicine. In these studies, pravibismane was tested against the biofilms grown from specific pathogens. In the blue arrow on the right side of the chart, you can see that pravibismane demonstrated a 6-log reduction of Pseudomonas biofilms at the same MIC for that pathogen. This is something that's typically not achieved by other approved therapies. And pravibismane also demonstrates anti-inflammatory activity via modulation of a key cytokine by over 200-fold. So this activity is anticipated to provide additional clinical benefit to our current indications and presents additional therapeutic opportunities for us. Now, as the saying goes, a picture tells 1,000 words. These are not pleasant to look at, but these are actual photographs from our phase 1b studies treating diabetic foot infections with topical pravibismane. In this study, we treated patients with chronic wounds that had a mean age of 6 months when they were enrolled in the study, meaning they had not healed using standard of care for 6 months. They were treated with pravibismane for 2 to 3 weeks. And on the picture on the left side, you can see the wounds when they came into the study. And then you can see in the second and third pictures, the wounds 2 weeks after end of treatment and 4 weeks after end of treatment, where you can see that they continue to close. While this was a safety study and not powered for efficacy, pravibismane did demonstrate a 3-fold reduction in wound size and 6-fold reduction in amputation rates compared to standard of care. Based upon this data, the FDA has said our next full study may be pivotal. We are currently doing a phase 2 study to further evaluate efficacy that has been funded in part by a $2 million grant from the US Navy. And in 2024, we plan to initiate a phase 2b/3 study, which, as I said earlier, may be pivotal. This presents the opportunity for potential FDA approval and commercial launch by 2027. We've also completed preclinical studies for inhaled pravibismane, where it's consistently well tolerated in GLP tox studies and in vivo studies. It also demonstrated in vivo efficacy against non-tuberculous mycobacterium, as well as Pseudomonas aeruginosa. In the chart here, you can see how inhaled pravibismane demonstrated a dose-dependent efficacy against M. abscessus, which is the most pathogenic and rapid-growing mycobacterium for which there is no approved inhaled therapy. We're targeting 2024 for initiation of phase 1 human studies for inhaled pravibismane. We are actively engaged in licensing discussions with pharma companies interested in pravibismane, and we're quite pleased to announce that we have a signed term sheet with a publicly traded pharma company interested in acquiring global licensing rights for topical pravibismane to treat diabetic foot ulcer infections and potentially other derm applications. Pravibismane's potential attracted backing from prominent strategic partners, including the Cystic Fibrosis Foundation, CARB-X, NIH, and the Department of Defense, amounting to over $21 million in non-dilutive grant awards. We are planning to raise $18 million to advance our current programs to very key value-driving milestones over the next 18 to 24 months. These include completing our current phase 2 proof of concept study treating diabetic foot infections, readiness for initiating a potentially pivotal DFI study, including the manufacture of our GMP clinical drug supply, completion of a phase 1 study for inhaled pravibismane in healthy volunteers, and readiness to initiate phase 2 studies. And we're also conducting additional studies on pravibismane's immunomodulatory activity. Kareem Lalji, our CEO, and Brett Baker will also be attending bio. And we're interested in discussing potential investors as well as potential partners for our other indications. Thank you so much for your time and interest today. I invite you to reach out to us to ask questions and discuss how you might get involved. Thank you.
Thank you, Stephen, and congratulations on the recent development. That's super exciting.
Thank you, Wendy.
So our next speaker is Dr. Carolina Vallant, CEO and CSO of Voxel Bio Innovation. Voxel is using bioprinting technology to develop human-like cancer tissue models for the drug development industry. Voxel's tissue models will identify the best drug candidates earlier in the drug development process, increase the predictability of drug behavior in humans, saving time and money for pharmaceutical companies. Over to you.
Thank you, Wendy. Hello, everyone. As Wendy said, I'm Dr. Carolina Valente. I'm the CEO and CSO of Voxel. And I want to tell you a little bit of the story behind Voxel and what we are doing with our exciting technologies. When we see the drug development process and when we start going down to understanding a little bit more, what we see is actually 95% of oncology drugs are actually failing to get to the market. And this failure rate is highly associated to how these drugs are being tested. They're being tested in very simplistic platforms in the lab. Then they are tested in animals and in people. And what we see is the results, they just don't match. Lab translation of data from the lab to all the way to clinical trials is actually less than 8%. So we are talking about an industry that is spending a lot of time, a lot of money, and drugs are just not making to the market. So what Voxel is proposing is a better drug platform to be screening these drug candidates. And that's exactly what we are doing using bioprinting. So we are creating human-like cancer tissue models using 100% of our own technologies. And these human-like cancer tissue models, they contain real human cells. And they contain a blood vessel-like structure. And the idea is that people will be able to inject the drug candidates through the vasculature of these tissue models. So let me show you here how this works. Our human-like cancer tissue models, as I said, they contain this 3D environment, and they are created by combining our bioprinter, our vascularization software, and our proprietary bioinks. What we have as a result is a 3D human-like cancer tissue model that contain real cancer cells. And the idea is that drug developers, pharmaceutical companies will be injecting the drug candidates through the blood vessel-like structure that we are bioprinting, and we'll be analyzing how these drug candidates are actually interacting with the cancer cells. The goal with these platforms is to be used as a screening platform, so we actually do not require FDA approval, but to be using it to eliminate the inviable drug candidates earlier in the drug development process. And because we have created 100% of our own technologies, we have a lot of flexibility on how to create the vasculature, how to create, how to mimic those cancer cells, and so on. Our resolution is very, very high. We are actually Canada's first high-resolution bioprinter, down to a resolution of below 1 micron. What that means is that we are able to create physiologically relevant tissue models using our technologies. And our goal with Voxel is we are aiming to put these tissue models on the market next year as consumables, and we're also aiming to commercialize our proprietary bioinks. That is basically our printer's cartridge. The goal with these bioinks is that people already using bioprinting technology, we made these bioinks for the first time fully universal, which means that it can be used in any other bioprinter out there. They won't be able to mimic the same tissue models that we are creating because of our printer and our software that's being kept in-house as a trade secret. So my background is in tissue engineering and bioprinting. Sebastian has joined us from Aspect Biosystems as our director of engineering. Kevin will be the one attending bio and our director of strategic alliances, and Evan is our lead of tissue engineering. Voxel was fundraising last year, and we raised $2 million, and this year we are raising an additional 1 million to extend our runway until summer next year. But our goal going to bio is really to gather strategic partners in terms of validating our technology on top of course, the fundraising. So the fundraising is mainly focused on our seed round that is going to happen next year, in which we are aiming to raise about $5 million. In terms of partnership, we are gathering beta testers for our BioInk. Our BioInks are currently under beta testing, but would like to have more beta testers. And we are also looking for strategic partners for validation of our tissue models. In terms of validation of our tissue models, we're really focused on CROs and pharma, but we also want to be using drugs that actually have failed in clinical trials to be using our platform to demonstrate that our platform has the potential to indicate that drug failure earlier on in the drug development process. So the goals with these partnerships is not just to use our platform as drug candidates, but we also want to be using drugs that have failed and engage in partnerships with that. What we see is the drug development process has been quite cemented for a while. This industry is changing recently. The FDA Modernization Act came to propose alternative in vitro platforms to animal models, and for the first time, bioprinted tissue models were referred as one of these alternatives. So there has never been a better moment to be exploring technologies like what we are doing at Voxel, and that's our goal. Our goal is really to accelerate life-saving therapies, getting to the cancer patients sooner, getting to the people that need this technology sooner. Thank you very much.
Thank you very much. It's great to hear your presentation. I just think it's so exciting what you guys are leading. It's amazing.
Thank you, Wendy.
Our next speaker is Lucas Donahan, Executive Director of Business Development at Zymeworks. Zymeworks is focused on developing novel multifunctional therapeutics for the treatment of difficult to treat cancers. Zymeworks' current pipeline includes antibody drug conjugates, ADCs, and multispecific antibody therapeutics, one of our leading antibody companies. So over to you, Lucas.
Thank you, Wendy. I hope you're seeing the correct slide view.
I'm seeing your slide and I can hear you perfectly.
Great, thank you so much. It's great to be here and on the panel with so many amazing companies. So I'm going to give an overview of Zymeworks today. We're really, as Wendy said, developing novel medicines for multispecifics and antibody drug conjugates, and really focusing on difficult to treat cancers and other serious diseases. So our goal really is to leverage our internal R&D engine, which creates these novel therapeutics utilizing our in-house platforms, to develop molecules that are either first or second line in their market or their target indication, that have a potential accelerated approval pathway, and hopefully be able to bring these molecules to the clinics pretty quickly in those indications that have the worst patient prognosis. Zymeworks has been around for about 18 years now, really started life as a platform company, generating novel platforms, first for multispecifics with our asymmetric platform, and then broadening that to include platforms for antibody drug conjugates, as well as then other platforms that bring in either a masking effect or other mutations to bring in multifunctions within different types of therapeutics. And we've validated this clinically with our lead molecule, zanidatamab, which is in registrational studies, and then our second molecule, which takes that same zanidatamab and adds an antibody drug conjugate to it, which is ZW49, now in phase 2 studies as well. I mentioned briefly what our pipeline is. This highlights zanidatamab, which we've recently partnered with Jazz Pharmaceuticals, as well as BeiGene, to enable the commercialization of this molecule and really make sure that this novel HER2 agent gets to the patients that need it the most, particularly in gastroesophageal adenomas and in biliary tract cancers, which are the first 2 pivotal studies that we have ongoing with this molecule. As we look to the future, we think about our preclinical pipeline. First, zanidatamab zovodotin, which is our ADC of that same HER2 molecule. That's in phase 2 studies now. We're starting phase 2 studies, and we're excited to continue to move that forward with our partner BeiGene and certain APAC countries. But then our next wave of molecules really start to leverage our more new and novel platforms that we've developed. In particular, topo-1 isomerase ADC platform, as well as then kind of a next generation T-cell engaging platform, with both of which we'll speak a bit more about as we go through the presentation here today. I'm going to get into the platforms now and first talk about our asymmetric technology for multispecifics. This is a bit of a busy slide, but it does really highlight the flexibility of the asymmetric platform to enable multifunctional therapeutics to kind of target any kind of targets that you want, but also not just antibody-based therapeutics, but digging into cytokines and Fc fusions, bringing in a masking component. Where we're really focused, at least initially, are T-cell engagers, and then what we're calling tri-T-cell engagers or tri-TCEs, which we believe are the next generation of T-cell engagers. And we'll talk about those as we go forward. As we think about T-cell engagers in that space, there really are a number of challenges that remain when it comes to bringing T-cell engagers to solid tumors. And we've tried to outline them here, where the first one is really a narrow therapeutic window and limitations due to cytokine release and toxicities that we've seen. The second would be low T-cell infiltration or T-cell energy, and finally, a immunosuppressive tumor microenvironment. And we've tried to develop ways that we can kind of attack both of these. The first one is a novel format, which I'll talk about in our ZW171 program. The other two are our kind of next generation platform, which bring in an additional co-stimulation component. So either for the low T-cell infiltration, we bring in a CD28 inhibition, or for immunosuppressive tumor microenvironment, we bring in a PD-1 or anti-PD-1 component to it. So it has 3 different areas that it's targeting. So for ZW171, this is our 2x1 bispecific mesothelin T-cell engager. And what we found as we developed this molecule was that the format really mattered. And so through our protein engineering, finding a molecule that really drove differentiation within its clinical space and versus the other clinical benchmarks, we were able to do that by identifying a format that had 2 scFvs targeting mesothelin, while one targeted CD3. And the scFv piece was really important versus a Fab to make sure that you're driving the right therapeutic window for this molecule. When it comes to ADCs, we have somewhat of a differentiated approach to ADCs, where we think about the entire molecule. And so while this highlights a lot of our payload capabilities, we also bring into it really developing a novel molecule holistically, including the antibody that really differentiates, as opposed to just throwing a payload onto an antibody that's already there. And that's really evident in our ZW191 molecule, folate receptor alpha, targeting topo-1 ADC. We spent a lot of time really identifying the right antibody for this application. And that means finding an antibody that is optimized for internalization, payload delivery, tumor penetration, not necessarily the strongest binder, but the best antibody for ADC properties. And then we used our new topo payload and linker system to conjugate to this molecule as a DAR8, which is also a bystander active molecule, and really believe that we're generating a best-in-class molecule here, even though the folate space is quite crowded at the moment, but targeting an IND in 2024. But we talked a little bit about designing fit for purpose ADCs. And today we're really focused on clinics that are already validated in certain unmet needs, but as we think about the future, going into more novel molecules, and so working with companies to define novel molecules, novel targets, thinking about ways that we can expand from monospecific ADCs into bispecific ADCs, and how can we better drive tumor selectivity that way, thinking about different payloads and conjugation methods, and then kind of what comes beyond the topo space. So we're really excited about our preclinical assets, and you can see certainly there's a number of molecules here that are targeting the topo-1 ADC platform. Our next two molecules, again, as we talked about, are 171 and 191, which we're moving pretty quickly towards an IND in 2024. And then following behind that are two additional topo molecules, GPC3 and NaPi2b, which we're excited to continue to move those forward as we think about INDs in the 24-25 timeframe. So I'll be at Bio. I'm excited to connect with a number of different folks, from VC companies as well as a bunch of other companies. We'll be talking about potential partnering opportunities, both in licensing and out licensing opportunities. Thank you so much.
Thank you, Lucas. Always great to get an update on what's happening with Zymeworks. So thanks very much. We look forward to you joining us in Bio with BC delegation. Our next speaker is Dr. Nicolette Hansen, Associate Director, Chinook Therapeutics. Chinook Therapeutics is a clinical stage biopharmaceutical company focused on developing precision medicines for kidney diseases. Their goal is to change the course of kidney care by making dialysis and transplant unnecessary for most kidney disease patients. Over to you, Dr. Hansen.
Thanks, Wendy. It's great to be here presenting today. Can you see the slides in presentation view?
Yes, I can hear you. I don't see it in presentation view though, so.
Okay, how about now?
Oh, yep, perfect.
Okay, perfect. Thanks. As Wendy mentioned, Chinook is developing precision medicines focused on kidney disease. I will be making a number of forward-looking statements today during the presentation. Chronic kidney disease is a severe and growing problem worldwide. About 10% of the global population suffers from some form of kidney disease. And currently there's limited therapeutic options to intervene in the disease course. So many patients progress to end-stage renal disease and kidney failure. There's a common misperception that if you have kidney failure, you can go on to dialysis or have a transplant. But it's really underappreciated how poor these outcomes are. If you're a kidney dialysis patient, you would need to visit a hospital or a dialysis clinic over 150 times a year. It costs about $200,000 annually to be on dialysis for a patient. In addition, the survival rate at 5 years is actually fewer than 50%. So this statistic is actually worse than patients with metastatic cancer. So really poor prognosis. If you're a patient that needed a kidney transplant and you were unable to secure a live donor, you could be on a wait list for over 4 years for a kidney. It costs $400,000 per transplant. Kidney transplantations, those kidneys only last about 10 to 20 years. So it's possible that you will need multiple rounds of transplantation in your lifetime, especially if you're a younger patient suffering from severe disease. So at Chinook, our goal is to really change this paradigm to intervene earlier so that patients can live and be treated for their disease so that they may not experience kidney failure and all of the poor outcomes that result. Historically, it has been challenging to develop drugs in this space for a number of reasons. First, kidney disease was thought of as a side effect of other diseases like diabetes. And secondly, the kidney itself is a really complex organ made of over 30 different cell types. However, we do see a change in the dynamics for developing drugs in the space. And we do believe now is the right time to be developing drugs for kidney disease. With the advent of the omics revolution and development of novel tools like single-cell RNA sequencing, there's certainly been an increased understanding of the underlying disease biology. And that's led to the identification of new targets and pathways that are key drivers to disease progression. And maybe more importantly, for the near future, the FDA is now recognizing surrogate markers like proteinuria and EGFR as registrational endpoints, making accelerated approval of new kidney therapies possible. Traditionally, clinical studies had focused on hard kidney outcomes. This required thousands of patients over many years to read out. And, in the kidney disease space where you're focusing on rare kidney diseases like Chinook is doing, that's just not feasible. So certainly the shift of thinking at the FDA, along with novel tools and data sets to better stratify patients, has allowed us to shorten that clinical development path from 5 years down to 2 to 3 years in much smaller targeted patient populations. And that has resulted in a number of approved drugs for kidney disease just in the past couple of years. In addition, there are over 40 late-stage clinical drugs in development. So really an exciting time to be in the space, an exciting time for kidney patients as new therapies become available. So Chinook is developing the leading pipeline in kidney disease therapies. Our lead program is called atrasentan. This is a small molecule endothelin receptor antagonist, currently in a phase 3 for the rare kidney disease IgA nephropathy. We do expect top-line data to read out in the second half of this year. Concurrently, we're also running a basket trial of atrasentan in other proteinuric glomerular diseases currently in a phase 2. And we hope to report interim data from these cohorts at major conferences and events this year. Bion 1301 is a monoclonal antibody that targets the APRIL pathway, also for IgA nephropathy. This program is in the phase 1/2 and has the potential to be truly disease modifying for IgA nephropathy patients. Last fall, we presented some really exciting and robust interim data from these cohorts, and that has made us comfortable with moving this program into a phase 3, which we plan to start in the middle of this year. CHEK336 is a small molecule LDHA inhibitor for treating a number of rare kidney stone diseases. We had recently voluntarily paused our phase 1 healthy volunteer study due to an idiosyncratic reaction experienced in one subject. We're currently interrogating and investigating this case and plan to provide an update in the future. In terms of a research and development pipeline, we have a really robust and multiple programs in other rare severe chronic kidney diseases based on internal development research programs, as well as collaborations and in-license opportunities. Moving forward, our goal is really to expand and build our pipeline. And we do that with internal research efforts augmented by business development activities. In terms of financials, we're in a good position. We have over 350 million in cash and investments as of March of this year. We do have global rights to all of our programs, except for in the Greater China territory, where we created a joint venture called San Reno, which has commercialization and development rights for our 2 lead programs, atrasentan and Bion 1301. We do intend to keep North American rights for all our programs. And so in terms of our partnering objectives, we are looking for a potential commercialization partner in Japan and other Asian countries, and are also exploring whether we will need a partner in Europe. As I mentioned in the last slide, we are working hard to build our pipeline, and so are open to collaboration discussions or new in-licensing opportunities. So with this last slide, I'd just like to thank Wendy again and the LSBC team for inviting me to present today, and the audience for your attention.
Thanks very much, Nicolette. Great overview. And we look forward to seeing you at Bio. Our next speaker is William Annette, CEO of Sustained Therapeutics. Sustained Therapeutics is a clinical stage company developing non-opioid medicines for the treatment of pain. The novel sustained release delivery platform has the potential to provide effective, long-lasting solutions for pain. Over to you, Bill.
Thank you very much, Wendy. And let me just share the screen. Can you see that?
Yep, I think you need to put in presentation mode.
There we go. That work?
Yep, that's perfect. And I can hear you perfectly.
Great. Thank you very much. Well, very happy to be here today and to tell everybody a little bit about Sustained Therapeutics. Here is our disclaimer slide. And I want to talk about three things with you. So first off, our initial market is very large, and it's the non-opioid pain management market. Secondly, the way that we are delivering our medication is a unique platform technology that can be used for multiple other indications. And finally, we're a clinical stage company. We have 2 phase 2 trials underway. So a quick note about the pain market. There's very high unmet need. Pain, obviously a major contributor to the opioid crisis. But there are not a lot of good alternatives to opioids. So we believe from a commercial standpoint that we have a very strong competitive advantage. We've done a lot of research with physicians, key opinion leaders, which is very important, of course, when you're in trials to know what your customers think. And we've got projected peak revenues of about $300 million US in our first indication and $1 billion in the second. So that's very exciting. The platform is interesting, again, developed at UBC. And so we are spun out of UBC. What has been developed is a novel locally injectable delivery system. Basically, you take a gel, a polymer gel, and inject it into the area of acute or chronic pain. We've got a very good patent position. We had US and Japanese patents issued and in national stage consideration across the world. And a lot of potential in the pipeline, not only in pain, but also in cancer and in various urethral diseases. On the clinical side, very successful phase 1 completed in 2021. We started a phase 2 trial in January 2022 in acute postoperative pain. And just in March, a month or 2 ago, we started a chronic pain trial. The chronic pain trial is happening across 4 sites in Canada. The acute pain is at the VGH and St. Paul's here. We've got a pretty rapid development pathway because of the nature of pain trials, and also a low development risk. The way that we are doing this is we have reformulated lidocaine, which is an approved generic drug. And because of that, the regulatory pathway is usually quicker and more certain for these kinds of medications. So a little bit more about the delivery pathway here. What's been created is a gel made of 2 biocompatible polymers. And as I mentioned, we inject into the site of the acute or chronic pain. And what happens is that as we inject here, as you can see in the demonstration, it forms a viscous gel in the body, kind of like a toothpaste. And over about a month, the gel hydrolyzes, turns into water, and the payload drug, which in our case is the reformulated lidocaine, releases. So it releases at a steady rate over about 1 month. Another interesting fact here is that we can tune the polymer so that it releases over hours or days or even a month or more. So a lot of potential for the administration of different kinds of drugs. We're starting with pain because pain is a very difficult medical condition. There are not a lot of treatment options besides the opioids.
And when we talk to the physicians, we talk to the key opinion leaders, they tell us, look, we're looking for 5 things. We want a drug which is non-addictive. It needs to be long-lasting. It's not something you want people to be taking every few hours. It needs to be effective against all 3 kinds of pain, because there are different kinds of pain. Non-toxic, and it should be easily administered. And our product meets all of those requirements. And that's unusual, because there's really nothing else out there or under development which does that. As you can see, our product lasts for a month from a single injection. It does address all the 3 different kinds of pain: chronic, neuropraxic, and acute. Very low toxicity and non-addictive. So we're very excited about the commercial potential because of this competitive advantage. We've got a lot of different indications in pain. Our first 2 on the left here, you can see we are projecting peak sales of $300 million US, because there's about 5 million patients a year with chronic scrotal pain in North America. And that gets us to the 300 million figure. Over 13 million patients with chronic pelvic pain. So we are projecting $1 billion plus annual revenue. And then there are many other potential pain indications, things like amputations, trigeminal neuralgia, and so on. And then on the right, you see the total acute postoperative pain market is about 38 billion. And we would capture, we believe, some section of that. So a lot, from a commercial standpoint, a lot of potential. In terms of the pipeline, the top 3 rows here are our pain trials. So we have, as I mentioned, 2 phase 2 trials underway. The chronic pelvic pain trial, we hope to begin before the end of the year. So we'd have 3 phase 2 trials in pain. And then the 4th row, upper tract urothelial cancer, is an oncology focus. So it's using a different payload drug. We'll be using gemcitabine. That is under development now. We hope to be in clinical trials, phase 2 trial, by the end of the year in this orphan disease. This is exciting as well because it's an orphan. There may be an opportunity with the FDA for accelerated approval. Management team, not our first rodeo. We've all been in the industry for a long time. Dr. Martin Gleave and Dr. Graham Boniface have co-founders of the company. Dr. Gleave, a well-known professor at UBC. He is head of the Vancouver Prostate Center. Very, very well-known researcher, received the Order of Canada for his research in cancer. Dr. Boniface, our Chief Operating Officer, has been very many years in the industry, and QLT and other companies developing therapeutics. My background, I'm a commercial guy, did my MBA, was at Genentech for many years on the finance and commercial side, and this is my 5th time as CEO of an early stage company. And we're also gathering a team around us of very qualified, very, very talented people. So we're excited about the team. We're excited about the opportunity. At Bio, Dr. Gleave is going to be giving a podium presentation about our company on Tuesday, June 6th at 11 a.m. room 104B, and we'd love to have you in the audience and listen to Dr. Gleave go into more detail about our clinical and scientific background. At Bio, we're going to be seeking investors and partners, and we're already filling up our dance card for that event. And at the bottom is our contact information. So again, thank you very much, Wendy, and thanks to all of you for paying attention.
Thanks very much, Bill. Super exciting. I'll try and make sure I attend the presentation live as well on June 6th. That's fantastic. I want to thank all of our speakers. As I mentioned at the beginning, this was a subset of our really strong delegation that is coming to British Columbia. So thank you, Nicolette, Lucas, Carolina, Stephen, Jonathan, Chris. and Bill, and of course, the Honorable Minister Bailey, who will also be with us. I'd like to thank our event supporters for our bio delegation. So the province of British Columbia and our networking reception event sponsors, Chinook Therapeutics and Invest Vancouver. As some of you have learned today, we have a very exciting group of companies that are coming. This is the entire list in front of us right now. So as I've mentioned, 40 to 50 organizations. We also have a link to the booklet where you can learn about the company, what their objectives are at Bio, as well as who the contact details are, of which you've met many of them today. So please take a look at that. If you are attending also, we will have a presence. Life Sciences BC will have a presence within the Canadian Pavilion, where we will be there throughout the conference to help you connect with our companies, both those that are in attendance and also for those that may not be there this year, which we can help connect you to. And with that, I'd like to thank you very much for attending our BC Bio Showcase. And we look forward to seeing you in Boston. And thank you to Claire Nelson, who has been running this event behind the scenes. See you in a couple of weeks.